In our last lecture, we talked about mitosis, which is shown on the left in the yellow box. Mitosis is a type of cell reproduction. They refer to it as asexual reproduction because the two cells that are produced, which they refer to as daughter cells, are genetically the same as the parent cell. Genetically, by that I mean that the DNA in the nucleus of the cell, where the chromosomes are, the genetic material, is each new daughter cell gets a complete set, and they are identical to each other and to the original parent cell. So that's why they call it asexual reproduction. This can be used for forming clones. If you have a cell that goes through mitosis many times, you form a big population of genetically similar cells. So mitosis is very important for things like growth and for tissue repair. But for producing offspring that are different from the parents, a different type of cell reproduction is used called meiosis. And in meiosis, the daughter cells are not the same as the parent cells. And one of the big differences is that they have only half of the original number of chromosomes. So, for example, in humans, we have 46 chromosomes in our body cells. After a cell goes through meiosis, there's only 23, half. So sometimes meiosis is referred to as reduction division because the daughter cells are genetically different and they have only half the DNA as the parent cell. Also notice for every cell that goes through meiosis, you get 4 daughter cells instead of just 2 at mitosis. So we're going to look at the process of meiosis today. Some vocabulary. There's a lot of vocabulary terms in this unit. Somatic cells, we'll use humans as an example, but this applies to multicellular organisms in general. Somatic cells are general body cells. Soma means body. So using you as an example, your liver, your lungs, your heart cells, your skin cells, those are somatic cells, and they have a full set of chromosomes, 46. You can actually obtain those chromosomes and display them. So you can look for abnormalities or missing chromosomes. That's called a karyotype. I'll show you one in a moment. So we'll look at that. So it's a display of all of the chromosomes in a cell. And one of the big topics that we need to understand before we understand meiosis is that of these 46 chromosomes, each one of them has a partner, a mate, I guess you could call it, that it goes with, sort of like pairs of shoes. So you could think of your cells as having 23 pairs of shoes, and they go together. So if you threw them in a big box and mixed them all up, you could still pull out, like, the right high heel and the left high heel, the right boot, the left boot, the right running shoe, the left running shoe, and so on. So those pairs of chromosomes that go together are called homologous chromosomes. Okay, so it's vital you understand what these are in order to understand meiosis. In mitosis, you didn't really even need to know what they are, because in mitosis, the cell just copies everything and the daughter cells get everything. But in meiosis, that's not going to be the case. So once again, homologous chromosomes are chromosomes that are paired together because they share some common traits. We haven't really talked about DNA or genetic material much yet. We will in this course, but basically homologous chromosomes carry similar genetic material. not the same, just like a right shoe and a left shoe aren't exactly the same. Homologous chromosomes are not identical copies of each other, but they do carry genes made out of DNA for similar traits. So, for example, if one homologous chromosome has a gene, a piece of DNA on it for eye color, its homologous partner also has a gene for eye color in the same place, but it might not be exactly for the same color. Like one might be for blue eyes and one for brown eyes. Or for blood type, one of your chromosomes might carry a gene for type A blood, and the homologous partner for the B marker on blood cells. So they're carrying similar but not identical traits. Or one more analogy is that I like to use with chromosomes are recipes. So homologous chromosomes are like cookbooks, and they have similar recipes, a recipe for lasagna and for chocolate cake and tomato soup and so on. So they're for the same dish, but they're not identical copies. They might be, different ingredients, different. They're for the same dish, but there's differences in them. And one more thing about homologous chromosomes is that you inherit one from your mom and one from your dad. So homologous chromosomes are always inherited one from each parent. And so sometimes they call them the maternal and the paternal chromosome. Okay, so look at this bottom line. Both chromosomes are carrying genes that are involved in the same inherited characteristics: blood type, eye color, whether your hair is wavy or straight, things like that. Here's a karyotype I was telling you about. These are actual human chromosomes that have been stained where they can, in a way that allows them to match them up into homologous pairs. So you see that they've taken these chromosomes from a human and paired them up with their homologous partners. So like, let's look, for example, at, chromosome number 9. Chromosome pair number 9. So these are homologous chromosomes. One came from your mom, one from your dad, and like I say, they're carrying similar genes. Now look at this pair down here. This is an unusual homologous pair, the XY chromosome pair. They are considered homologous, but actually in males, which this is a male, the X chromosome, which is the bigger one, and the smaller Y chromosome, they do go together, but not as homologous chromosomes, but not because they carry identical genes. They do not. But they pair up in a different way in that together they determine genetic sex. So humans that inherit an X and a Y chromosome are genetically male. Humans that inherit 2 X chromosomes are genetically female. So they are considered a homologous pair as well. So why am I going on and on about homologous chromosomes? Because homologous chromosomes during meiosis are going to first pair up. So they're going to find each other in the cell and actually synapse or pair up. And then they're going to be split up into separate cells. So by the end of meiosis, every cell has only one of chromosome pair number 9 and one of chromosome pair number 6 and so on, and the X or the Y, but not both. So that's how you read. So when I say that meiosis is about reducing the number of chromosomes by half, I'm not talking about just any random half of the chromosomes. I'm talking about one of each homologous pair is going to go into each daughter cell. So by the end, the cells that are produced by meiosis have no more homologous chromosomes. They've been split up. That doesn't happen in mitosis. We'll talk more about this in another lecture, but since we were talking about karyotypes, I thought I'd at least briefly mention for now how they obtain the chromosomes, or one of the main ways they can obtain the chromosomes to do the karyotype, and that is during pregnancy, the fetus is in an amnion, a sac of fluid, and some of their cells, their skin cells, get sloughed off into that amniotic fluid, and they'll just take a sample of that fluid, and in there's going to be some of the baby's skin cells, and then they can obtain the chromosomes from that. So that's called amniocentesis. Again, we talk about it more in another lecture. Okay, some more vocabulary in red here. So the sex chromosomes I mentioned, those are in humans called the X and the Y chromosome. You do need to know females are XX, males are XY. And so those are, again, the sex chromosomes. So you just have one pair of those. And then the other chromosomes are called autosomes. So those are, all the other pairs, 1 through 22. Autosomes and sex chromosomes. Okay, some more vocabulary. A cell that has a full set of chromosomes, like 46 in humans, is called diploid. Di means two, and they call it that because there's homologous pairs. The chromosomes are arranged in pairs of twos, and diploid is also designated as 2N. Okay, you definitely want to know this when we get to genetics because they use this a lot. So your general body cells produced by mitosis are diploid. Well, I don't see the other term, but if they have only half, so that would be the next slide. If they only half of the number of chromosomes, that's called haploid, Haploid, and that's designated with an N. So there's haploid and diploid. Okay, so here we see a cell with some chromosomes floating around in it. These cells have already been through the S phase of the cell cycle, and that's something, by the way, that was covered in the mitosis lecture, so make sure you know what happens in the S phase. This is before mitosis or meiosis occurs. All the chromosomes are copied, so there's 2 copies of them. Sorry, I keep advancing. 2 copies, they're called sister chromatids. They are identical. Okay, but if you look through this cell, you can probably figure out what are the, I'm so sorry about that, homologous chromosomes, because they usually make them the same size, like big, medium, and small. So these homologous chromosomes are going to have to pair up. And the reason they have to pair up is so that you can then split them up. Because how are you going to orderly segregate them into different daughter cells if you don't first put them together so you can line them up and split them up? So let's look at the process of meiosis. By the way, maybe we should first talk about the overall function of meiosis. So the reason we want to make unique cells that are different from either parent cell is that so that these cells from meiosis, which are called gametes, and in animals we call them egg and sperm, these gametes will fuse with gametes from the opposite sex to form a new individual that's diploid, called the zygote, and that zygote is going to be genetically unique, from the parents. So meiosis is all about is a type of sexual reproduction in which the offspring are different from the parents. You are not a clone of your parents. You have half of one parent's DNA, one set of homologous chromosomes, and then the partner chromosomes are from your other parent. So meiosis is about producing the gametes, the egg and sperm. Gametes is another name for the daughter cells that result from meiosis. And here's that term I was trying to tell you about in the previous slide, haploid. See, egg and sperm have only 23 chromosomes, which if you look at this picture, you can see why that's necessary. They each need 23 chromosomes because when they fuse during fertilization to form the new zygote, which is diploid, you need to restore the 46 chromosome number. That's the characteristic number for humans. So you can't have gametes with 46 chromosomes in them, because if they were fertilized by another gamete with 46 chromosomes, that you would double the number of chromosomes in the offspring, and every generation would double the number of DNA, and that's not going to work. So meiosis is about producing gametes for sexual reproduction. Okay, here's some main differences. Let me just get them all up here between meiosis. Meiosis, I didn't even tell you this part. Meiosis is divided up into two stages, meiosis 1 and meiosis 2. And each one of those has the same 4 stages that mitosis has. So they each have a prophase, metaphase, anaphase, telophase. So there's like prophase, metaphase, anaphase, telophase part 1, and then prophase, metaphase, anaphase, telophase part 2. So really 8 steps instead of 4. And you definitely need to know them all for lecture and even for lab. On your lab final, there'll be some questions about this. So meiosis 1, the first stage, is all about separating the homologous chromosomes into 2 separate daughter cells. So that's the reduction stage, where you're going from diploid to haploid. And then meiosis 2 is really very similar to mitosis. You are splitting up the sister chromatids, just like you do in mitosis. The only difference here is that you're starting with only half the number of chromosomes, a haploid number, because the homologous chromosomes are already have been separated into different cells. So they're not around in meiosis 2. They're already in different cells. Okay, so this is not showing the details of all the steps, but it's kind of giving you the big picture. Here's 2 chromosomes. These are, I'm so sorry about that. These are, this is a homologous pair. Okay, and normally you just have 1 copy of each, but in the S phase of the cell cycle before mitosis, you got to copy each one. So now they have sister chromatids here. But this still counts as 2 chromosomes, even though they're duplicated. And then during meiosis 1, notice the homologous chromosomes get separated into different cells. And then in meiosis 2, the sister chromatids are separated. So, and then you end up with the 4 at the bottom, 4 haploid daughter cells. Okay, so now we're ready to go step by step through the different phases. So I'm just going to focus on the main events that occur in each one of these. Okay, so once again, we're starting out with the chromosomes in the duplicated form. The duplicates are called sister chromatids. They're attached by a centromere. Here's the picture from the last lecture, just to remind you. Here's the 2 sisters. They are genetically identical. There's the connector centromere. So that's what we're starting with. 46 of those, using humans as an example. Okay, so here's the 4 phases of meiosis 1. Prophase 1 is the first one. Lots of things happen in prophase 1. I guess the easiest place for you to start is just to first learn mitosis, and then know that in prophase 1 of meiosis, the same things happen that happened in prophase 1 of mitosis. The nuclear envelope breaks down. The chromosomes condense, and the spindle apparatus forms from the centrosomes, the centrioles, the microtubules that are going to help move the chromosomes around. Okay, so that happened in mitosis too. Nuclear membrane breaks down, the chromosomes get more compacted, and the spindle forms. Okay, now here are 2 things that happen that do not happen in mitosis. The sister chromatids, or the homologous chromosomes find each other. They synapse with each other, that's what it's called. So they come together and synapse. So here we see 3 homologous pairs that have synapsed. Sometimes they call them tetrads because if you count sister chromatids, there's 2 plus 2, 2 in each duplicated chromosome, so there's 4 of them, hence tetrads. So that's prophase 1. And then one other thing's going to happen that's not shown in this picture. I'll show you in a moment. It's called crossing over, where there's some swapping of genetic material between the homologous chromosomes in a tetrad. So we'll review that in a moment. Prophase is complicated. Here's metaphase 1, where the tetrads, I like to call them the synapsed homologous chromosomes, line up on the equator. And then in anaphase 1, these synapse breaks and the homologous chromosomes separate. They go to opposite poles. And then this cell is going to pinch in 2, and that's going to be the end of meiosis 1. It pinches in 2. That phase is called telophase, and cytokinesis is the pinching in 2 of the cytoplasm. So let's look at some better pictures here. Here's more vocabulary. Synapsis, that's what the term used for the homologous chromosomes finding each other and sort of being knitted together temporarily. So they call it a tetrad. And you don't really need to worry about that term. That's what they call the connectors that hold the homologous chromosomes together. Okay, now crossing over is an exchange of genetic material, and that's shown in this picture. So here's a homologous pair of chromosomes, one blue, one red, and this is happening in prophase 1, they're going to swap some genes, some recipes, some genetic material, and so look at what you get in the end. It's called crossing over. You've got some genes from one of the chromosomes got swapped over the other, and vice versa. Now, I don't want you to think that these chromosomes are lacking anything at the end. So, for example, if this is a cookbook, using that analogy, each homologous chromosome still has a recipe for lasagna and chocolate cake and tomato soup, but they might have swapped some of their recipes, like they might have swapped their chocolate cake recipe. So they still are complete. Each one has a complete set of genes that's carried by that chromosome or that homologous pair, but they've just mixed it up some. This is what meiosis and sexual reproduction, it's all about genetic variation, producing genetically unique offspring. That's because that's an advantage to our species, or any species, to have genetic variability. If everyone's a clone of each other, the species is in danger of, they're coming, I usually use the example of a virus, but, some kind of climatic change or a virus or fungus or something that, if it affects all the members of that species and they're all the same, then it could wipe out the whole species. Whereas if you have genetic variability, it protects the species as a whole. So that's what meiosis is all about. So that's crossing over, swapping of some genetic material between the homologous chromosomes. Okay, so in this slide we see the homologous pairs now lined up on the equator. So this is metaphase 1, which is a pretty simple phase. But while we're looking at this, let me just go back for a moment and read and list for you all the things that happen in prophase, because I want to make sure I'm getting that across. So the nuclear membrane breaks down, the chromosomes condense, the spindle apparatus forms, the homologous chromosomes synapse, and crossing over occurs. It's a lot. Okay, here they are now lined up on the equator, and then that's metaphase 1. And then in anaphase, the tetrads are going to separate and be pulled towards opposite poles of the cell. So that's what you're going to see, and that's another vocabulary term. That's called disjunction, separating the two. So here you see that in this third picture here. The homologous pairs are being pulled to the north and south poles of the cell. And then, not shown, but this is going to be followed by telophase and cytokinesis. And that cell will become 2 daughter cells, each with now only 3 chromosomes. So in this example, the full number of chromosomes is 6, and after meiosis 1, it's going to have 3. It's always half. This is telophase 1. So the maternal and paternal homologous chromosomes reach opposite poles, and now the cell pinches in 2, and we call those haploid cells because they have only half of the diploid number. Okay, and then we have meiosis 2, which has, noticed, the same 4 phases: prophase, metaphase, anaphase, telophase. And this one is quite similar to mitosis. So look carefully at this picture over here. Oh. I'm sorry, I have problems with the cursor. So at the end of meiosis 1, here are the 2 daughter cells that resulted from meiosis 1. So notice, look carefully, they each have 3 chromosomes in them, and neither one has homologous chromosomes in it anymore. Okay, now even though these chromosomes are in the duplicated form, you call these cells haploid cells. See how down at the bottom it says haploid? So they're haploid. You don't count sister chromatids, you count unique chromosomes. So each one has 3 chromosomes. It's haploid. So each one of those daughter cells is going to go through meiosis 2. So prophase 2 of meiosis is just like mitosis. The chromosomes recondense, the nuclear membrane, depending on how much of it forms, is going to break down again. The spindle will form again. The chromosomes will line up on the equator of the cell. And then this time the centromeres break and the sister chromatids are pulled to opposite poles during anaphase 2 of meiosis. And then you have telophase, where they've reached the opposite poles, and see how the cell's starting to pinch in 2, cytokinesis. That's going to occur, and you're going to get 4 haploid daughter cells. And in this case, they're not duplicated anymore. There's just 1 copy, not 2. So if this was happening in a male human, these would become 4 haploid sperm, or they could become egg cells in females. Although, as we'll see later in females, cytokinesis, where the cells pinch in 2, is unequal, not equal, so you end up with 1 huge egg and 3 tiny little, they're called polar bodies. We'll talk about that later. So that's meiosis. It's good to start with mitosis because it's simpler, and then learn meiosis. So again, this is comparing them. So we see mitosis results in 2 diploid, genetically identical daughter cells. Meiosis results in 4, for every cell that goes through it, 4 haploid gametes, or daughter cells. And they're unique. They're even unique between each other because of crossing over. So here's a little exercise to compare them, comparing mitosis and meiosis. So when do the chromosomes get copied? In mitosis, they get copied during. I'm sorry, I'm having so much trouble with the cursor. During interphase, the S phase, and there's only 1 division. One prophase, 1 metaphase, 1 anaphase, 1 telophase. There is no synapsis of homologous chromosomes or crossing over, and you end up with 2 diploid daughter cells, and the functions are mainly for growth and tissue repair. Okay, what about in meiosis? When does the DNA replicate? Same time, during the S phase of interphase. Remember, S stands for synthesis because the DNA is going to be copied. How many divisions? 2. There's prophase, metaphase, anaphase, telophase 1, and then prophase, metaphase, anaphase, telophase part 2. So there's 2 divisions. Synapsis and crossing over does occur during prophase 1. And the daughter cells are 4, and they're haploid, and none of them are identical to the parent cell. And the role is sexual reproduction to produce gametes. So here it is filled in. So, this is definitely going to be a big topic on your next test. Lots of questions about mitosis and meiosis. What happens during metaphase 2 of meiosis? What happens in anaphase of mitosis? When are the chromosomes duplicated, and so on. All right, so one more thing I wanted to mention is just to kind of review and all the different ways that meiosis, that genetic variation, in other words, is the genetic variation occurs. So mutations in the DNA are the ultimate source of genetic variability. When DNA gets, for whatever reason, when the structure of it changes, your genetic makeup changes. So mutations are the ultimate source of different variations of genes, like the gene for blue eye color versus brown, or type A blood versus type B blood, things like that. And sexual reproduction with meiosis allows us to reshuffle, like shuffling a deck of cards, to reshuffle all of these mutations to come up with genetic variation, which I already mentioned is an advantage for species because it improves the chance of survival of the species as the species environment changes. There's a bigger chance that at least some members of that species will be able to survive and reproduce. So here are the 3. We've talked about the first 2 already, but these are the 3 main mechanisms that contribute to genetic variation during sexual reproduction. So crossing over, swapping of genes between homologous chromosomes, that happens again in prophase 1 of meiosis. Independent assortment of chromosomes. Okay, this happens, and I have a picture of this coming up. This happens in metaphase 1 of meiosis. So we'll talk about that. And then random fertilization is another mechanism for genetic variation. So what's independent assortment? Well, this is crossing over. Okay. Independent assortment refers to the random arrangement of non-homologous pairs of chromosomes at metaphase 1. So at metaphase 1 of meiosis, you have all these non-homologous chromosomes lining up. Like in this picture, you've got the big and the little ones. Those are non-homologous pairs. Like a pair of flip-flops and a pair of boots. Those are non-homologous. So the way the non-homologous chromosomes line up is random chance. So maybe, like you see on one side, maybe all the reds are lined up on one side and the blues on the other, or maybe they're mixed up. It's random chance. But the way they line up is going to affect which gametes they end up in. So because they can assort independently of one another on the metaphase plate on the equator of the cell, that leads to even more chances for variability. It's not, in other words, it's not like all the maternal chromosomes, the ones that had been inherited from the mother, say, line up on the right side and the paternal ones on the left. It's not organized like that. It's a random alignment of non-homologous chromosomes. So that leads to a huge number of variations. Okay, so they call that independent assortment. And then random fertilization refers to which sperm fertilizes the egg. So in this picture you see a huge egg cell and a bunch of little sperm. And only one of those can fertilize the egg if it does get fertilized. In other words, the egg has 23 chromosomes and the sperm head has 23 chromosomes, and only one sperm can donate its 23 chromosomes to the egg to form a new zygote or baby. So which one of all these sperm is going to actually fertilize the egg is random. So again, this increases the genetic variability. They call it random fertilization. So crossing over, independent assortment of chromosomes of non-homologous chromosomes at metaphase 1, and random fertilization are three mechanisms for increasing the differences between offspring and thus genetic variation. So that's meiosis. So once again, make sure you understand mitosis first and then tackle meiosis. And do learn it well, and it will help you when we get to genetics, which is the next topic we're going to be covering.